institutions developed by this trust have created a niche in healthcare education, not only in hinterland of Mangalore, but also across the globe. In recognition for this contribution to the education system, in the year 2008, the Ministry of Human Resource Development, on the recommendation of the University Grants Commission, conferred the deemed to be university status. Yennapoya University thus became the first unaided deemed university to be established in the Dakshina Kannada district of Karnataka. The university, following the accreditation in 2015 by the National Accreditation and Assessment Council with an A grade, has diversified its academic prowess and scope by establishing Yennapoya Pharmacy College and Research Center and Yennapoya Institute of Arts, Science, Commerce and Management in the year 2017, the Yennapoya Ayurveda Medical College and Hospital and the Yennapoya Homeopathy Medical College and Hospital in the year 2018, the Yennapoya Institute of Allied Health Sciences and the Yennapoya College of Naturopathy and Yogic Sciences in the year 2021. Yennapoya University has secured accolades by various national and international certification and accreditation agencies. The significant ones being consistent ranking within 100 by the NIRF for 3 consecutive years. ATL ranking of institutions on innovation achievements by the Ministry of Education on 2 occasions under self-finance or private institutions category as an endorsement for the innovation and entrepreneurship development efforts among students and faculty of the university. The university has also been ranked with 4-star rating in KSURF Young University category in 2019. In recognition of contribution to the new knowledge generation, the university has been ranked among the top 50 institutions in India in the SCI MA Geo Institutions ranking consecutively during the last 2 years. There are multiple faculties of study in the university which are represented by the standalone constituent units. These institutions are spread over a total built area of 1,44,453.11 square meters to accommodate academic and administrative activities. Curricular diversification has been the focus of the university. It offers more than 100 programs at the undergraduate, postgraduate, super specialty, doctoral and postdoctoral levels. The university has enrolled more than 6,000 undergraduates, 600 postgraduates and doctoral students, complemented by over 500 well-qualified teachers trained in ICT-enabled teaching-learning processes. The classrooms, seminar halls and demonstration rooms are ICT-enabled with Wi-Fi facilities extended to all strategic locations in the campus. The integration of information communication technology in teaching learning meets innovative educational standards. Self-directed learning is supported through e-resources on the customized learning management system. Choice-based credit system incorporates a wide spectrum of electives in majority of the programs offered. Internships, industry visits, community postings, skill development training and early exposure to research equip the students professionally competent for employment. The mentorship program supports the mentees throughout the duration of the program to enhance learning experience and personal growth. Community-based training is imparted through the urban rural health centers, PHCs, Rural Healthcare and Development Center, and mobile clinics to inculcate social responsiveness. The university library caters to the learning needs of the students and faculty. It has a vast collection of books, manuscripts, ancient books, digital learning resources. The library transactions are fully automated, facilitating accession and lending of books. The digital library, which is available at all the constituent units, helps the students and faculty to access the e-resources. All the academic departmental laboratories are well equipped for providing training. To enhance experiential learning and skill acquisition, the university has created Centers Advanced Surgical and Skill Enhancement Division (ASSEND), and a state-of-the-art Advanced Comprehensive Clinical Training and Simulation Center. These facilities are one of their kinds in the country and provide training support to the students and faculty from other institutions. The examination section is fully automated with an indigenously developed examination management system procedures involved in the pre-examination, examination, and post-examination processes. This has enabled to bring in several technological reforms in the examination system, enabling speedy announcement of results. The university has been making constant and concerted efforts to actively engage in research. Research support facilities available in the university include state-of-the-art central research facility with modern equipment for conducting advanced research in emerging areas like microbiomes, cell signaling, nanoscience and microfluidics, stem cell and tissue engineering, systems biology and molecular medicine, omics technologies, bioinformatics and biostatistics supported by highly qualified manpower, funding, informatics tools, Animal House and Ethics Center. The Ethics Center has the unique distinction of imparting NIH USA funded postgraduate academic programs in research ethics. The Center for Nutritional Studies fosters nutrition-related activities in India. The policy and advocacy has led to government's initiative in launching a direct benefit transfer scheme for all TB patients to enable access to a better diet. This center has developed maternal nutrition advocacy, implementation, policy and planning in collaboration with Ministry of Health and Family Welfare, UNICEF, National Center for Excellence and Advanced Research on Diets. Mpa Technology Incubator aimed towards identifying, fostering and mentoring innovators, entrepreneurs in the healthcare innovation process. Recognized by the PIRAC, Mpa Technology Foundation supports startups and more than 60 patents have been applied, published or awarded. The university has signed more than 150 MOUs with several national and international institutions of excellence for research, faculty and student exchanges. The 4 hospitals cater to the teaching and training activities of medical, dental, Ayurveda and homeopathic disciplines. In addition, there are yoga, naturopathy and wellness centers provide treatment and training facilities. The Medical College Hospital is NABH accredited with 1,100 beds with the latest facilities for robotic surgery, organ transplantation, 3D MRI, modular operation theaters, ICUs, and emergency services. The diagnostic laboratory is NABL accredited, covering services of biochemistry, microbiology, pathology, and transfusion. The Allamilu Charitable Foundation, supported by Tata Trust in association with the Mpa University, is establishing a comprehensive cancer care center. Medical College Hospital provides free food, free and subsidized treatment to all the patients. The hospital has received several awards from government and non-government agencies for its outstanding contribution to public service and workplace safety measures. The university provides equal opportunities to all students, upholding the principles of equity. The observance of cultural fest, religious festivals, NSS activities, and disabled-friendly and barrier-free environment provide the opportunity for students from the diverse background to effortlessly blend with the campus life. The international student cell caters to the needs of overseas students to seamlessly adapt to the campus environment. The Student Council provides a platform to undertake various co- and extracurricular activities. The university provides scholarships, fee waivers, and fee concessions for financially weaker students and promotes gender equity. Career Guidance Cell prepares the students for entrance competitive recruitment examinations. The Placement Cell, in association with Alumni Association, organizes campus placement drives. Students participate in extension and outreach activities through camps, awareness programs, and have adopted several villages for comprehensive healthcare support. The university has created excellent sports and auditoria facilities to support the physical and recreational requirements of the students and faculty. Endurance Zone, the indoor auditorium has a multi-purpose hall, basketball court, shuttle badminton, volleyball, gymnasium, squash court, table tennis, and board games. The outdoor sports facilities include international standard grass soccer ground, cricket practice nets and stadium, kabaddi court, throwball court, and athletic stadium. There are 13 hostels for students, 7 for women and 6 for men, and are provided with solar water heating, uninterrupted power supply, and Wi-Fi facilities. The centralized kitchen, Foodcraft, caters to the food requirements of students, staff, and patients, along with 2 canteens and 15 cafeteria spread across the campuses. The laundry services, Laundry Line, in the university campus has capacity to handle 5 tons of clothes, is a boon for students, staff, and patients. The campus has several environment-friendly operations which include rainwater harvesting, water conservation, waste management, energy conservation, and promotion of alternate energy utilization. As an endorsement for these efforts, the university has been recognized and awarded by prestigious international, national, and state bodies and is amongst the top 400 institutions ranked by Times, the Global Impact Ranking, which mainly focuses on achieving sustainable development goals. The university has adopted multiple UGC programs to sensitize and inculcate social responsiveness among the students and cross-cutting issues like gender, environment sustainability, human values, health determinants, right to health issues, emerging demographic changes, and professional ethics. Commitment to our nation and society is embodied in telemedicine initiatives, relief activities during environmental disasters and the COVID-19 pandemic outbreak, and support to cleanliness initiatives of the Government of India. Yenapoya, deemed to be university, provides an inimitable milieu for holistic growth and upholds all functional integrity of a markedly progressive institution. the accreditations and laudable transformations in its overall performances over the years are ample evidence of Yenapoya's contribution to education. Through its futuristic vision, it intends to transform into one of the acclaimed educational institutions. The strategic plan Yen Vision 2035 emphasizes
Good morning, Natasha. You can start.
Good morning. Yes. A very good morning to everyone present here. I, Dr. Natasha Naval Agarwal, Assistant Professor, Department of Pharmaceutical Chemistry, YPCRC, welcome you all to the 5th day of the one-week Faculty Development Program on Recent Trends in Pharmaceutical Sciences, organized by the Staff Welfare Committee, YPCRC, in collaboration with APTI. On behalf of Yenapoya Pharmacy College and Research Center, Yenapoya deemed to be university, I would like to extend a very to our speaker of today's session, Dr. K.M. Nurullah. I also welcome the delegates attending from all over the country and abroad, and my dear colleagues. A hearty welcome to all. Before we proceed, I request all the participants to drop your queries in the chat box, which will be later conveyed to the speaker after the session. You're also requested to fill the feedback form after the session. Moving forward, it's my privilege to present the biosketch of today's speaker. Dr. K.M. Nurullah completed his Bachelor of Pharmacy and Master of Pharmacy in Pharmaceutical Chemistry from the Tamil Nadu Dr. Mpa Medical University with distinction, where he was the college topper as well as the department topper. Sir further completed his PhD from College of Pharmacy, Madras Medical College, Chennai in the year 2016. He worked as a lecturer in Bapatla College of Pharmacy and as an assistant professor in Sri Balaji College of Pharmacy, Andhra Pradesh, India. Sir then worked as a researcher for a period of 5 years from 2012 to 2017 in College of Pharmacy, Madras Medical College, Tamil Nadu. He also worked as an associate professor in the Department of Pharmaceutical Chemistry and Analysis in Deccan School of Pharmacy, Hyderabad. Sir is currently working as an assistant professor in Department of Medicinal Chemistry, College of Health Sciences, RC University, Ethiopia. Sir has guided 4 MPharm students and his research interests include design and synthesis of novel selective small molecules therapeutics for the treatment of various infections and inflammatory diseases, development of new synthetic methodologies, target-oriented synthesis, computer-aided drug design, and analytical method development and validation. Sir's area of expertise include multi-component synthesis, separation and purification, GCMS, LCMS, QSAR, Pharmaco4 Modeling, and docking. Sir has also numerous publications in various national and international journals and has conducted multiple scientific sessions as a resource person. With this brief introduction, I would like to invite Dr. K.M. Nurullah to take over the session and enlighten us on the topic, an overview on Pharmaco4 and its modeling. Over to you, sir.
Sir, please unmute your mic, sir. Sir, Dr. Nurullah, sir, please unmute your mic.
Madhu, Vanusali, Allah Rasul, Mama, Good morning to one and all. Thank you, Dr. Natasha Agrawal, for your brief introduction. With that, I would like to welcome all the participants for today's session. And without any further delay, I would like to go for the session. Can anyone confirm that I am audible and my screen is on the display?
Sir, your voice is echoing, sir.
Okay, 1 second.
Kindly check your network, sir.
Is it fine now?
Sir, now it is fine. Natasha, you can unmute your mic.
Is my screen?
Yes, sir. Your screen is visible, sir.
Yes. So today's session is like an overview on Pharmaco4 and its modeling. So we all know about computer-aided drug designing. Once again, Pharmaco4, once again, if you see, it is one among approach in the computational.
Sir, sorry to interrupt. Your voice
Yes.
is not clear, sir. And I request you, sir, for a full screen, sir. It is still in a slide model.
Okay.
Hello?
Yes, sir.
Yes, sir.
Is it full screen now, ma'am?
Sir, kindly display your slide, sir. It is in full mode only. The slide is minimized.
Now?
Kindly click on your slide, sir.
Yes, is it clear, ma'am?
No, sir. Your slides are not visible, sir.
Okay. Now, ma'am?
No, sir. Yes, sir. It's visible now, sir.
Visible now? Okay.
Yes, sir. Yes, sir.
Okay. I'm audible and the slides are visible. Hello?
Yes, sir.
Yes. Can I start, ma'am?
Yes, sir. Please, sir.
Yes. Okay, for today's topic, an overview on Pharmaco4 and its modeling. Generally, Pharmaco4 modeling is one interesting technique on computer-aided
Excuse me, sir. Sir, your voice is echoing, sir. And could you please make your slides as full screen? Your voice is echoing, sir.
These are all natural models, chapter models. These are the tablets from application of natural models. So she is facing.
Sir, kindly click on slideshow, sir, which is on your screen.
Yes, ma'am.
So that she is facing the.
Sir,
So if you are
It's in
giving
slideshow mode,
important
madam.
drugs to that means you will be
Not visible.
it has minimized, sir.
Minimized.
this is the advantage of natural
Sir, in a present, kindly click on A tab, then you can go, sorry, your entire screen, then you can show your slides, sir.
They don't know whether it's from some other practice. No, they have act. These are the extracted step preparation. These are not authentic. They power and conversion of any drug in the tablet form is very easy. So we can convert it
Okay, now, ma'am.
Okay, sir, you can continue, sir.
Prepared from different sources of
One second.
plants.
Proper now, sir. It is proper now.
Those interactions due to the interaction of different kind of molecules.
Is it in slideshow or full screen?
Now she's hospitalized. So what they are giving you so how many days you on this? Past 5 days she has to.
Sir, your screen is proper, sir. You can continue with your talk, sir.
Okay. So, Pharmaco4 modeling is one among interesting approach in computer-aided drug design. So, once again, today's session, based on the time constraint, we are not going to cover entire different types of approaches in Pharmaco modeling, but I will try to.
Sir, sorry to interrupt. Your screen is not visible, sir.
One second, ma'am. We'll join another.
Screen is not visible.
One second. Is it okay now, ma'am? Is it okay,
No,
ma'am?
sir, not visible.
Now is it okay?
We can see, sir. The slides are visible, sir.
Okay, ma'am.
Almost all the plants, whatever they are having.
Should I start, ma'am?
Right now. Yeah, all right.
Okay.
Yes, sir, you can start. And I think your some other system is on, so there is an echo when you are talking, sir.
Okay. Yes, straight away we will enter into that. Actually, for 15 minutes, I already wasted. So, disclaimer. For today's presentation, there is a small disclaimer. So, this presentation is for education purpose only. Some of the contents pictures are taken from the web articles and some tutorials for teaching purpose only, and its respective authors own their copyright. So, all opinions, suggestions, comments in this presentation are on my personal capacity only. So, for today's session, content agenda is we will be having slight flavor on what is computer-aided drug designing, and followed by Pharmaco4 modeling introduction. Again, Pharmaco4 modeling, as I told, it is a vast area.
So your
There are
voice is
many
not clear,
approaches.
sir.
Recently, there are updates on many of the approaches. So here we will be discussing a very easy and very important approach as a demo session. Then the easy
Sorry,
and
sir.
very important Pharmaco4
Excuse
modeling
me, sir.
approach. What I was to tell is common fit based Pharmaco4 modeling. It will be a
Excuse
demo.
me, sir. So your audio is not clear, sir.
Is not clear?
So what kind of range you are in that time?
Ma'am, now, ma'am?
Now, okay, sir. Please carry on, sir.
Yes. So the common feature based 3D Pharmaco4 modeling. Today we will be having a short and sweet demo. The very important thing here today is it will be all the softwares or web servers what we are going to use for today's session will be in open source platforms. So each and every servers softwares which we are going to use all are from open source platform. So all the participants need not to worry about the subscription, commercial package cost, all those things. So each and every person can, in day-to-day basis, they can even practice this Pharmaco4 modeling. So finally, there will be a important area like I would like to show the web link for CAD
Sir,
software
your slides are not moving, sir.
all at one pool. So I hope this session I will not bore you all. Be interesting, and at the end of the session, I believe that all the participants can having a very good exposure on one of the Pharmaco4 model techniques. So to that, computer-aided drug design. So drug discovery and development is an expensive and complex process. This we all know, and minimum there will be more than 10 years of efforts for a successful drug to enter into a market. So because this in these 10 years, there are many challenges task, and there is a involvement of multidisciplinary approach. So CAD methods generally they think that CAD methods are very easy as well as it can shortcut the efforts both in ways of time and economical wise. So where you will place this CAD method? Generally at the drug discovery process, early
Excuse me, sir. So
stage.
your voice is breaking, sir. And the slides are not moving.
Sir, you can switch off your camera. Only if your audio you are using, I think it will
Okay.
improve.
Okay.
And then the slides are not moving also, sir.
Okay.
Still, it is in the first slide.
Now? Okay, ma'am.
Sir, the voice is still breaking, sir.
What slide, ma'am? What about the slides, madam? Is it CAD slide?
So the slide, it's the first slide itself, so it's not moving. So the slides are not moving.
Now, ma'am.
No, sir. It's still the first slide, sir.
Sir, you have to go in the presentation mode for your entire screen, sir.
Yeah, it is in entire screen, ma'am. The presentation mode is in entire screen.
Okay, sir. I hope it is in kindly. Okay, sir.
I don't know what technical issue is here. Internet is okay. Internet is okay. Still the problem exists, right? Now what is which slide?
So we can't see the slides, sir.
Is it now clear?
So, yes, sir. Now it has. So, can you just try moving the slide, sir?
Getting moved.
So the CAD slides, yeah, visible. Yes, sir. It's visible now, sir.
Okay.
Yes, sir.
Visible now?
Yes, sir. But your voice is breaking, sir.
Sorry. Maybe the echoing. Echo. Maybe. Is it okay, ma'am? Now?
Yes, sir. Yes, sir. Please continue, sir.
So the CAD methods generally will be employed early to mid stage of drug discovery processes. This are connected much to the drug discovery process with rapid advance in computing capacity, data storage, softwares, and algorithms. So CAD has applications in target fishing. So if you don't know the target, even through CAD, we can just identify the targets, target validation, identification, and selection of the lead and its operation. So there are many types of these approaches in CAD. So generally, we used to know that structure-based.
Sir, your voice is still breaking. Can you share your PPT with us so that we'll help too?
Ma'am,
You please share your slide with us. Sir, please share your slide with us. We'll present from here and you can talk so that this problem will not happen.
Okay. Okay. Ma'am, I have sent. Still. Ma'am, can you tell now? Is it visible? I mean, change the connection.
Yes, sir. It's visible, sir. It was visible, sir.
Yeah, I will proceed, ma'am. This.
Yes, sir. Yes, sir.
If anything trouble means you just tell me.
No, sir. Now everything is clear, sir.
Clear. Okay.
Yes, sir.
Sorry for the interruptions, my dear friends. Pharmaco4 modeling. So just a concept of Pharmaco4 was first introduced in 19th century by Paul Ehrlich, who defined the Pharmaco4 as a molecular framework that carries the essential features responsible for the drug's biological activity. So Pharmaco4 modeling is not new to us. In 19th century itself, the term Pharmaco4 has been introduced. Likewise, Toxico4, Metaphor4 are all there. So after a century's development, the basic Pharmaco4 concept still remains unchanged, but its intentional meaning and application day to day have been expanded considerably. So if you see, according to the definition of IUPAC, a Pharmaco4 model is an ensemble of steric and electronic features that is necessary to ensure optimal supramolecular interactions with a specific biological target and to trigger either block its biological response. So based upon the IUPAC definition, if you see, a Pharmaco model is nothing but assembly of steric and electronic features, which is very, very important to ensure the supramolecular interactions with the biological target. Once the ligand or anything which is going to bind, its response can be either way by activating or blocking the response. So if you see, in a very simply understanding manner, a Pharmaco4 is a description of molecule features which are necessary for a molecular recognition of ligand by a biological macromolecule. A model which we generate generally explains how structurally diverse ligands can bind to a common receptor site. So if you see, Pharmaco4 models can be used to identify novel ligands. Say, if you have a map or a model, Pharmaco4 model, through that model, we can just identify novel ligands either by de novo designing or virtual screening, which will bind to the receptors. So now you see a general Pharmaco4 modeling approaches. So there are two types, ligand-based and receptor-based. Please don't confuse with this receptor-based with the structure-based drug discovery. This receptor-based is once again an approach of Pharmaco4 modeling only. Receptor-based Pharmaco4 modeling, otherwise known as structure-based Pharmaco4 modeling. Here it is purely ligand-based Pharmaco4 modeling. So once again, if you see each type has two further divisions, like ligand-based 3D QSR method and ligand-based 3D Pharmaco4 modeling without QSR. Here receptor-based, once again, is structure-based. Receptor knowledge you will know. The difference here is even the structure knowledge or receptor knowledge, if you don't know, you can perform the ligand-based Pharmaco4 modeling. If you have a known structure, receptor structure, then also you can perform a Pharmaco4 modeling based upon the geometry of active sites present in the receptor. Once this model has been developed, as you see in the display, the model has been developed through its novel ligands can be identified through virtual skinning by cross-checking with the large number of chemical databases are available. You can cross-check, and through de novo designing by fragment linking, etc., etc., and other applications like lead optimization, multi-target drug designing, activity profiling, target identity, etc., etc. So now, as I said, that ligand-based Pharmacopoeial modeling once again is of two types, structure-based once again of two types. Now here, if you see the first type, ligand-based 3D Pharmaco4 modeling, it's similar to hip-hop type. Okay, so today's session is with this approach we are going to discuss common feature-based hypothesis. The interesting thing here is it just looks the compounds having structural homology. That's all. Common features and a narrow activity range. In this method, like a traditional or conventional, conventional Pharmaco4 modeling, we no need any activity data for this type of Pharmaco4 modeling. Only if you know that this ligand or this drug or this molecule is going to have activity on towards that receptor, or for any handful of disease, this drug is going to work, means what you can do is just you can collect the drugs. Minimum three to four compounds is sufficient for a common feature-based hypothesis like hip-hop type. So just you are going to combine and you are going to overlap and check the structural common features in the present in that. Then based upon that, the model will be generated. It will be very simple, very easy technique. Then next technique is like ligand-based 3D QSR Pharmaco4 modeling. It is hypogen generally. It generates hypotheses that are able to estimate the activity of molecules quantitatively by using biological analysis data. So for this type of Pharmaco4 modeling, you need to have biological data in the form of any like IC50, LD50, or any MICs like that. Any biological data is required for this type of hypogen modeling. So it allows the correlation of structural activity data and the biological activity for Pharmaco4 modeling. Here in this 3D QSR Pharmaco4 modeling, you need a diverse set of ligands like actives and inactives. You can make a highly active compound, moderate active, and even inactive compounds, so that there will be a statistical orientation based upon the structure as well as the biological activity. And you might have heard about this type of 3D QSR Pharmaco4 in daily basis. But the common feature hypothesis, if you see, means that that deal is very simple, and you can even practice very regularly in day-to-day research. So next, if you see structure-based Pharmaco4 modeling, it's once again a Pharmaco4 modeling technique where you will know the exact structure. You will have a 3D receptor structure. So the protocol of structure-based Pharmaco4 modeling involves analysis of complementary chemical features of the active site. The binding pocket site details you will be having, and based upon the active site features, their spatial relationship, and active amino acids idea, you can derive a subsequent Pharmaco4 model assembly with these features. So once again, here I told you there are two types. It is like macromole ligand based SBPM, as well as another type is macromole structure based pharmacophore. The difference is there you will have a native ligand in that. A native ligand in that native ligand will be helpful for having insight on your pharmacophore model. Another model type is like only macromole you have. It will be a native ligand absent type receptor, so that you can perform by assessing the active site informations. So for today's session, we will be discussing about hip hop type, that is common feature based hypothesis. So just we used to tell that Pharmaco4, what is Pharmaco4? We have just we saw in the earlier slides. It is like the fragment which is important for pharmacological response, or any type of affinity. After affinity, there will be a response that can be carried out by the Pharmaco4 fragment, the fragment present in the molecule. Likewise, we have Toxico4 and Metabophore, which are respectively responsible for their processes. Here, in 3D Pharmacophore modeling, like 1D we have 2D. Now here it is common feature based is hip-hop 3D Pharmacophore modeling. We have some typical pharmacophore features. So these are the features we will be looking for a set of molecules which are having narrow activity towards a common receptor. So these typical features will be looked for that, and the alignment will be taking place and model will be generated. So what are they? Hydrophobic centroids. Like if you see hydrophobic centroids, generally it is like a methyl group, ethyl group, any hydrocarbon material, fragment. Then we have aromatic rings. Then we have hydrogen bond acceptors like OH, NH. Like then hydrogen bond donors are there, like hydrogen bond acceptors like oxygen and nitrogen. Hydrogen bond donors, if it is like OH, NH. Then we have ionizables like ions like positively charged and negatively charged, like cations, anions, respectively. Then now, based upon our session, I will tell you the flow of how you can perform a common feature-based 3D Pharmacophore modeling. Just first, what you are going to do is you need to select ligand or approved drug molecules, minimum 3 to 4, which binds to a common receptor sites. That is very important. Here you can see it's my suggestion, like you can go for a clinically approved drug molecules. A handful of diseases are there with known drug molecules in the market. So instead of going for a published ligands, which we have some doubt on its bioactivity, so better if you can go for FDA approved drug molecules on which has a narrow activity towards the common receptor sites. Like you can go for a minimum of 3 to 4. Note this point, like common feature based, even if you have 3 to 4 compounds, you can generate a model. But in 3D QSR Pharmaco4 modeling, like Hippogen, I told now, for that at least you need a handful of compounds, like at least minimum of 15 compounds is required for that type of modeling. Here for this minimum 3 to 4 compounds, advice is to go for a clinically approved drug molecules, FDA approved, or genuinity with the biological activity, genuine compounds you can go. Very important is that set 3 or 4 compounds what you are going to choose. The minimum is 3, 4. You can go up to 15, 20 also. That's not an issue. The important thing is it should bind to a common receptor site. Then next step, we are going to generate Pharmaco4 generation. So there are many softwares, web servers present for generation of Pharmaco4 model. Here I have in the slide, you can see PharmaGist web server. So as I told you, for this session, for this type of Pharmaco4 modeling, I am going to discuss all with open source softwares so that everyone can practice. So PharmaGist web server is a freely available web server for Pharmaco4 detection. It is generally ligand based. It does not require the structure of target receptor. So if you don't know the receptor, knowledge or anything, if it is not at all required for this pharmacophore detection. Only thing, the input is a set of structures. As I told, 3 to 4 is sufficient of drug molecules that are known to bind to common receptor. Here for this web server, your molecule should be in a mole-2 format. Okay, mole-2 format. So you might have heard about mole format, MDL, SDF formats. This web server accepts in the mole-2 format. Generally, mole-2 format is nothing but a file type which can record multiple entries. Like it can have information of molecule, bond, atoms, substructure, sets, 3D coordinates. Even if protein is there, it can have that information also, and any substructures are there, and also it can have. So these everything can be accommodated in this mole-2 file format. So this require mole-2 format type of input. Once again, I will explain how to convert the mole-2 also. Then if you have 3-4 compounds, as I told, it all should be in a zip file format. Don't go for a RAR format or any format, only zip format. It should be compressed to a zip format, and simply input, enter your mail ID, your result will be in minutes. So now, after you generated your Pharmaco4, what you are going to do is simply you need to visualize how the generated Pharmaco4 looks. Excuse me, can anyone tell that I'm going smoothly or not?
Yes, sir, it is running smoothly.
Yes,
yes. No network issue or anything. So your slides have
Pharmaco4 visualization software. So what you are going to do, the PharmGist will give you the generated model. So what you need to do is just you visualize what it is, what are the typical features, as we already discussed, whether hydrophobic is present or aromatic is present, acceptor, donors, what the feature is common and which is responsible for Pharmaco4. That information you can see, including its geometry, distance, everything, etc. So what you can do, you can once again you can visualize. So for this you can use the BioDiscovery Studio visualizer. So generally BioAva Discovery Studio is a commercial software. But visualizer for this platform is freely available. You can download. The link is there. After the session, I have already shared the slides to the management. I mean, they can give this slide so that links are there. You can download. So BioAva Discovery Studio Visualizer is free. It is full of feature-rich molecular modeling application for viewing, sharing, and analyzing protein and small molecule data. So next step, what you are going to do, very important step, you have generated a Pharmaco4. Then now you want to do a virtual screen. What you can do is the exact geometrical feature of the Pharmaco4 model, what you have generated, will be just searched across the databases. So database, many databases are there, chemical databases are there. Here I'm using Zinc Farmer. Once again, it is a free database, chemical database. Millions of compounds are there, which can be having drug likeliness property. So what you can do is searching for diverse chemical compounds with which share the same features as of your generated model, arranged in the same relative orientation. So it is a free Pharmaco4 search software for screening the purchasable subset of Zinc database. Generally, its updates occur monthly. So the obtained Pharmaco4 feature from PharmGist can be loaded and searched across the Zinc database. So now what you can do is we are going to do a demo on this process. So for this short and sweet demo, I have taken COX-2 inhibitors, selective COX-2 inhibitors, non-steroidal anti-inflammatory derivatives. Like we have 4 drugs. I have chosen 4 drugs for this: celecoxib, etoricoxib, rofecoxib, valdecoxib. So these all are having a binding specificity towards COX-2 inhibitor. So it is already a well-known drug to all. So what I'm going to do is these 4 drugs I'm going to take. Already these all are FDA approved drugs. So in these 4 drugs, what common features are there that we are going to see, and we are going to generate a Pharmaco4 model from this, and we are going to search in the Zinc database. So for this, what we are going to do is I will just for the time convenience, I have already downloaded all these 4 molecules in SDF format. Can you see this screen?
Yes, sir.
Yes, yes. So you can see SDF, SDF, 4 SDF files are there, 4 drugs. These all are I have downloaded from the PubChem database. If you know, you can just, if you have Chem 3D software, anything, you can draw in mole format. After that, you can convert it into mole to format. So how you are going to convert it into mole to format? I have already converted it, but I will show you how to convert. So Discovery Studio Visualizer, I told you, it is not only useful for visualizing your model. You in this BioWire Discovery Studio, you can even convert any type of chemical format into any type of chemical format. So now one chemical type I will just convert and show you, so that you can do like that. Even I will open the PharmaGist software. So already we are running late. Simply PharmaGist. So you can see the screen here. PharmaGist web server is there. This is for Pharmaco4 detection. So once it is opening, it is very easy. No need of anything. Just click chemistry type. So it will ask whether you are not a robot. Simply just click it. Go to the server. Go to the server. So the page looks like this server page. So now, meanwhile, we will just this is the look of BioWire Discovery Studio Visualizer. So what I told, just we will be converting into SDF to mole to format. So what you can do, open. So it is under this your selected folder. Here SDF file. What I am going to do, Silica XDF file I am going to import. Just it is imported. Just you can click this. Yeah, that is dragged down. You can see this is the SDF standard structure that format, data format. You can see this I need to convert into mole to format. So you can just scroll if you need. Whatever you want, you can do in this molecular window. So just I want to save it. Save us. I will save it in desktop. Okay. Before saving, you need to do one thing, like you need to clean its geometry. Very important, because it is raw data. If you draw also, its geometry cannot be perfect. Even if you download from some databases, its geometry will not be perfect. So better you can clean its geometry and then say structure clean geometry. Just it will clean its geometry after optimized it. Then save as. Here it is showing at the Discovery Studio files. You can just change it into Sybil mole to file. Sybil mole to file. Just simply save. It will be converting into mole to format. This is I have saved in desktop. You can see this. It is in mole to file, mole to format. So like this, what 3 or 4 molecules you have, 10 molecules, you need to just clean the geometry. You need to save it in mole to file. The second step is you need to make a zip file, that is compressed zip file. So what I am going to do is, so mole file, I am going to select all the 4 molecules I have selected. Simply I am going to archive it. Right click. Add to archive. So you see here, you can change the name, whatever you want. I'm going to keep XYZ. Already I have COX inhibitors. Then see that active format, no need of R. R is not it is not recognized in the PharmaGist. So very important, it should be zip format. Zip format, XYZ, just click. It will be there. It will just create, and it is now here. It is zip format XYZ, that 4 molecules are there. Even you can just open and check whether 4 molecules in the mole to file format is there. Now simply you can upload into the web servers. This is the PharmaGist. Now we have gone web server. So just upload input molecules in mol2 format. Just choose the file, your XYZ. That's it. And enter your email address. I'm here entering this email address. Just submit. So why it is 12 compounds? Once again, check. Okay, I have selected a wrong thing. 1 second. So 1, 2, etheric oxide and cerebral. These 4 I need to select. I have to archive, zip, let it be like this. Okay. So now it is in a webinar. Now I will upload this. So you can choose the file. Enter the molecular, mail ID, submit. So now you can see these four drugs are there. It is validating. By the time it can give you the result also in your mail ID, email ID.
So we want a drug which is having this kind of toxicity should be less. So the synthesis of new and effective anti-conversion drugs become a huge platform, you can say, for ongoing research. So there were so many receptors, you can say the neurotransmitter that act in case of the epileptic drugs. For example, we are telling about the GABA potentiation actions. GABA is an inhibitor neurotransmitter, but is having the potentiative actions. Voltage gated sodium channels, some drugs, and some specially T-type calcium channel inhibitors we are also using. And Mpa receptors. Our target is our Mpa receptors where that particular quinazolone nucleus will bind. Mpa receptor, it is actually alpha amino 3 hydroxy 5 methyl 4 isooxazole propionic acid receptors. It is actually inotropic receptors. Basically, it is a glutamate. It is a glutamate receptor. Glutamate is 2 types. One is the inotropic, one is the metabotropic. So AMPA is inotropic receptor, which is an excitatory postsynaptic receptors. Today, I will not focus about the details about the pharmacology because it is very huge. So it is not possible within one specific hour time. So I will basically concerned with the chemistry part, but just in brief introduction, I am telling about the Mpa receptors. Our target for the quinazolone nucleus is the Mpa receptors. Although some sort of quinazolone nucleus, they are targeting the GABA, but mainly they are targeting the Mpa neurotransmitters. And this Mpa neurotransmitter is one of the non-NMDA type of neurotransmitters, and it is an excitatory postsynaptic transmitters. So we have to target that AMP receptors through the various ligand molecules. There were very less drugs are there, AMP receptor antagonists, very less drugs. So recently, so many scientists, they are continuously going for the research. I have found so many papers, very good papers, Nature as well as from the PubMed. So many research are going on about the quinazolone, about the AMP receptor antagonists. But I have found very less drugs are there, which are clinically approved as an AMP receptor antagonist anti-epileptic drug. So one of the drug is the perampanel actually. So perampanel, it is one of the drugs, pyridone derivative drugs. That perampanel, it developed during the screening research for non-competitive Mpa receptors. It is actually US approved drugs in the year 2012. It is under the schedule class 3 controlled drug substance for a potential drug actually. And the mechanism is that, it is bind to the non-competitive Mpa glutamate receptor antagonist effects. And successful models, it has been found that particularly the scientists have tested it in various models like your MES seizures, subcutaneous PTZ induced seizures, usually the 6 model like the kindling seizures. And this perampanel also used in case of the partial seizures. And recently I have found one article that is one of the decanoic acid one drug which is having the synergistic effect with the perampanels. Another drugs, telompenol, it is also one of the drugs, but it is having the clinically approved. It is not clinically approved. It is under the clinical trial still now. Because it is under confused, you will not get a good paper on the telompenol, but it is so many papers have write about that, but not it is clinically approved. Only the telompenol is clinically approved. It is a 2,3 benzodiazepine derivative. They bind to the negative, your non-competitive side of the Mpa receptors. They bind to the non-competitive side of the Mpa receptors, which are having the clinical trials for chronic indications, for example, epilepsy, sclerosis, as well as the malignant brain cancers. So this is about the telompenol, and it inhibits the Mpa receptor at the 10 to the minus 6 m concentrations. Next drugs. Is the GYK1 52466 and another 3 CP526427, CP465022. These are the very rare drugs which are used as an anti-epileptic drug bind to the Mpa receptors, and you will not get the good standard quinazolone derivative drugs. GYK 52466, it is one of the drugs. Another drugs is CP526C. This one is your quinazolone derivative, as well as CP465022. This is also quinazolone derivative. These two specific drugs, quinazolone derivative, which are having the anti-epileptic effect, they bind non-competitively to the Mpa receptors and having the anti-epileptic effect. Okay, and telompenol also is one of the benzodiazepine derivatives, so it also having the Mpa receptor antagonist. So why we are going for the quinazolone derivative? Due to this only, because very less drugs are there which are having identified, or you can say some are clinically approved drugs, but other drugs are on the experimental basis. So many good paper you will get, they are mentioning about the how they are inhibiting on the experimental models like MES seizure, pentylene tetrazole induced seizures, like that, but it is not clinically approved. The main problem associated with is that I am telling about the neurotoxicity, high amount of neurotoxicity levels, as well as the low protective index values. So our target is one of the attempt to go for a good quinazolone nucleus, which is having the anti-epileptic activity in case of the experimental models, as well as some Mpa receptor antagonism activities. So you already know about the quinazolone nucleus. We have already so many marketed available drugs, quinazolone derivative drugs. So the established drugs, you can say, for example, I am telling about the prazosin, which is in quinazolone derivatives. So this quinazolone nucleus has huge, you can say the biological activity, you can say the broad spectrum of activities. But our target is anti-epileptic because till now it is under the experimental basis. We are not getting any kind of clinically approved drugs. So today I will tell about the how you can modify the clinically approved drugs and how you are going for the structural modification of different binding sites to the quinazolone. Here actually you can see all kinds of paper they are showing that the binding to the 3 positions, the structural modification occurs to 2nd position, 3rd position, 4th position, 6th position and 7th position. 2, 3, 4 and 6, 7. They have just modified, they have just given some sort of structural modification like binding to different groups, and this particular groups having the tremendous biological effect. But the problem is that there is having the neurotoxicity value. Anyhow, we have to go for different modification. So that part I am telling. So these are the different, you can say the clinically approved quinazolone nucleus like methaqualone, mecloqualone, afloqualone, mebroqualone. These are the different CNS active neuropharmacological active drugs. These are the quinazolone derivative. Some methaqualone also it is also bad. It is not used because of the serious side effect associated with that. But depending, because of activity is good, but the problem is the side effect, that is why it is not used properly. But these are the various quinazolone nucleus which are having the different neuropharmacology, you can say anti-epileptic activities. So what is our research work? In which way you can go for the modification? What I have done in one of my research work, that part I am going to share. So this is one of my research work about the quinazolone nucleus I am doing. So basically, I have told that quinazolone is one of the huge biological activities. So huge biological activity means you can say any kind you will get, like your anti-cancer activity, CNS activity, some sort of antimicrobial activity, some immunomodulatory activities also. So this quinazolone nucleus, marketed available drugs are there in case of your anti-tumor activity, CNS activity, and some antimicrobial activity. But very, very less drugs are there due to the side effects. Our target is we have to see one quinazolone nucleus, but we can go for the different sites. The problem with the research laboratory with the faculties is that they are not giving sufficient time for the synthesis, or you can say the activities. So you have to segregate yourselves, you have to segregate the works in which way. One is the synthetic part, one is the characterization part, one is the activity part. So you go for the various structural modification continuously and synthesize the compounds. After synthesis, you go for the characterization. So after characterization means you can say the physicochemical characterization, all are part of the research. Design is one research, synthesis is one research, characterization is one research, and pharmacological activity is one research. But the problem in India is that we are compiling everything at a time. So that is the most peculiar thing we are doing. That is why we are not getting a particular good research. And at last we are going for the paper publication. So undoubtedly we are having the manipulation of data. But if you go for the research in particular section, that will be better. Whether it is designing, you can say the molecular design, or you can say the homology modeling, any kinds, or you can say the autodoc, you are doing drug designing studies or dockings. Then come to the synthesis part. So synthesis part, you just see the different, how you can go for the modification of the particular heterocyclic ring. Then we are going for the characterization like physico-chemical characterization, any kind of characteristic properties up to the 2D, 3D NMRs, recent development in the NMR studies. So that part you can do it. Whether it is UV, whether it is IR, whether it is NMR, whether it is mass. Then we are going for the pharmacological sections and evaluations and docking study that I have put here. So this quinazolone nucleus is having the biological effects. Basically, research is going on the two. One is the anti-cancer activity, or you can say anti-tumor. That is the non-small lung cancer therapy, breast cancer therapy, as well as the activity. This anti-tumor activity, recent advance is going on in the nucleus. So you, my dear faculties, you put on that quinazolone nucleus these two, whether it is anti-tumor activity and the CNS activities. So that two part you can analyze, go for various synthetic scheme and try to go for synthesis. But it is difficult to go for the 3 to 4 step synthesis. Very 2-step synthesis you try. 2 steps, 2 to 3 maximum is, but you try to modify the various structures of the quinazolone nucleus, different sites. For example, I have told 2, 3, 4, 6, 7 position you can modify. So that is one of the, see, we are going for the various research. One is having the synthesis, another is the structural activity relationship according to the biological activity and molecular mode docking. So this part will be the focus. Your target will be like that. Synthesis, molecular docking, synthesis, biological activity, as well as the structural activity relationship. Depending upon your biological activity, you have to make one conclusion about the SR, or you can say the structural activity relationship. This part in India we are basically doing. So this is one of the pictorial quinazolone scaffold as an anti-epileptic. I am telling all of you, just go this one of the designing and one of the synthesized compounds and having the activity, anti-epileptic activity. So I have gone through the various research articles, and from that I have put one conclusion in that particular scheme of the scaffold. You can say that different groups are present. So in the quinazolone nucleus, see, there are double bond N, first carbon atoms containing double bond groups. So 1, 2, 3. So 1 position nitrogen and 3rd position nitrogen. See, there are 2 nitrogens are there. So the double bond N nitrogen is the electron donor group. NS group is there. At the 3rd position, there is attachment of the M1 NS group to the any position of the aryl ring. So this one is also electron donor group. And see the electron withdrawing group here. Electron withdrawing group is the carbonyl compound C double bond O. C double bond of the position is at 4. The C double bond of electron withdrawing group, Another is the hydrophobic sites. So hydrophobic pocket, that means the aryl rings, that may be aryl distal array, you can say. So which way this particular electron withdrawing group or electron donor group or the hydrophobic group, they are having the activity. Generally, the electron donor group, that means see here there is presence of the lone pair of electron in the nitrogen. So this lone pair of electron in the nitrogen, it take part in the bond formation whenever you are going for the drug designing or you can say the docking studies, ligand receptor binding interactions. So there will be the hydrogen bonding interactions. In case of the drug designing study, we'll see the basic things in the hydrogen bondings. More in the hydrogen bonding, then you can say the predicted activity will be good. That we are thinking. Similarly, in case of the ammonia, it increases the solubility, it is basicity, increase the solubility. It also take part in the good biological activity. It is already in the experimental basis, it is proved by the various literature sites. Electron withdrawing group like your carbonyl group, C double bond O or C double bond S. These are, you can say, the carbonyl group. Their electron withdrawing group increases the, you can say, the stability as well as the anti-epileptic activity. In case of the hydrophobic sites like your aryl rings, that aryl ring may be ortho-para substitutions. Ortho position or para position, you can go for any kind of substitutions. For example, you can put hydroxy, methyl, methoxy, NH2 group, nitro group, or you can say the fluorine, chlorine, bromine, any kind of substitution. So my dear faculties, you can go for any kind of substitution in this aryl ring. If you go, then you will get the particular result. Definitely you would get the result because it is experimentally proved that this kind of quinazolone scaffold is having the anti-epileptic activity. See, anti-epileptic activity and the CNS depressant activity both are correlated. So if you go for the acute toxicity studies also, you will see in the acute toxicity study, the locomotor activity, you will get the one of the significant locomotor activity. If the locomotor activity, you will see that it is depressed, so it is having the huge CNS depressant activities. You will get that. So this is one of the quinazolone scaffold which is having the anti-epileptic effects. Another quinazolone scaffold, you see there is presence of hydrophobic aryl ring, same things. The hydrogen bonding domains like NH groups, the methylene spacers, the carbonyl compounds. See this particular group, the spacer, hydrogen bonding donors, hydrogen bonding domains, these are essential for the anti-epileptic activity. It is also experimentally proved. Similarly, the electron donor group, another position double bond N, this also very important, and the distal aryl ring, it is also approved. Distal aryl ring and the methyl spacers, these are having anti-epileptic activity. If you see that methaqualone, this is also having some sort of spacers and the aryl ring. That is why it is one of the experimentally proved anti-epileptic drug designing, and it is already synthesis and activity is there. So this kind of scheme, this kind of scaffold you can put, and you can go for any kind of changes. Suppose I am telling at the quinazolone nucleus, second position, third position, fourth position, sixth and seventh position, you can go for any kind of substitutions. Any kind of substitution means, for example, here it is given in the second position how the hydrogen bonding domains and electron donor group is there. You can put any kind of, instead of the aryl ring, you can put the heteroaryl ring like that. And in the fourth position, if it is having the ketonic group, you can go for the substitution other group also, which is having the good electron withdrawing capacity. So that is also having the activity. So you just try yourselves in the laboratory and try to synthesize the various compound. And the problem with the designing in the synthesis is that we can go for the enormous amount of designing lead molecule through the virtual screening process. But whenever it is come to the reality, but it is very difficult to synthesize the compounds. Very difficult to synthesize the compound because optimization problem will be there. Optimization problem with the solvent, optimization problem with the temperature, optimization problem with the timing, the particular means climate or environment, it is very difficult in the your academic institutes. So you go for the at random synthesis of various functionalities, but you have to see the structural activity relationship by observing the various literatures. Then only it is possible. So for example, I am telling, I have gone for the Autodoc, one of my studies. Autodoc I have done, one of the research center in Northeast, Institute of Science in Advanced Studies. So there I have done in the you can say there is one department. So in that particular department, I forgot that particular area. There were so many different sections in that. So in that particular pharmacophore group, so that they name it, the department is the pharmacophore group they have named. So in that group they are having the pharmacoinformatics. So in that pharmacoinformatics, huge these softwares are there. So they have told, I have also surveyed what you are doing. They have told that we are doing the virtual screening. We are going for the virtual screening of the various lead molecule, more than 1 lakh or 2 lakh. They find out. But the unfortunate situation is that whenever it comes to the reality in the laboratory, it is very difficult to synthesize. That is why the molecules are discarded. So my dear faculties, you just focus on the synthetic scheme, those who are specially the expertise in that particular field. You have to go for the synthesis by structural modification. Just see the structural activity relationship. For example, I am telling about the methyl. If it is methyl, then you can go for the long chain structures. If it is methoxy, that is also you can put it. If it is amino derivative, for example, one of the research is that the quinazole nucleus and amino group, if it is present, then it is having the huge anti-epileptic activity. And you can go for the modification with the NH2 group, in which way, suppose you can go for the hydrogen hydrate group, or you can say the phenyl hydrogen moiety. So that part you have to see. Or you can say the aminos like that, N dimethylamine, N diethylamine, like that you go for continuous searching, continuous synthesis. Then only you can get the results, that because synthesis part also one of the research. So that you can go and characterization. But until unless you are getting the good biological activity, it is nothing. That is the people thinking. But my thinking is that if you are going for the synthesis also, you are going to optimize a new synthetic scheme, solvent optimization, temperature optimization, addition of the different functional group, cyclization, converting the particular group into a new heteroaromatic compounds, derivative. That is also one of the research. So continuously go through that, continuously go to the labs, and you just synthesize the various schemes process. Then definitely you would get some sort of results. Very, very small you can do it. So next is, that is also one of the target compound that we have done. So you just see like the methaqualone, it is having the rings, aflaqualones. So these are the A is one of the compounds which is having the bromine substitution. Although bromine is having the toxic effect, but whenever it bind, bromine is one of they are the halogen groups, and they are the electron withdrawing group also. They are having the more electronegativity. So in that halogen, they are showing the good biological activities. As well as the substitution, the yellow color part, substitution of the substituted aryl rings. So these are showing the good biological activity. So coming to the conclusion, the target compound is that you just see the hydrophobic domain, small anionic group. So fluorine, actually here it is substituted the fluorine group. Basically the fluorine is means having the good stability, it is safe, and it is showing that the fluorine group having the good pharmacokinetic profiles. If you consider one of the antibiotic, I think you have already studied the fluoroquinolone antibiotics. The fluoroquinolone antibiotics, for example, I am telling the ciprofloxacin or ofloxacin, there is also having the fluorine derivative. If you go through the structural activity relationship, it has been found that fluorine is having the good pharmacokinetic profile. It adds to the good pharmacokinetic profile of that particular heterocycle or the antibiotics. So that is why it is also one atom that how it enhances the pharmacokinetic affinity of that particular nucleus. So you can put any kind of halogen substitution here, and at random you go for the synthesis. But it is very difficult to join. Very difficult to join the particular drug. Very difficult to join the different groups. So that part you have to go through the research, continuously going through the synthesis. So this is one of the target for the anti-epileptic research. Another research you just see here how the quinazole nucleus at the third position, it is having the different spacers, you can say, whether it is ethyl or methyl or butyl or isopropyl or isobutyl, like that. You can go for the structural modification. And the Y, you can put the NH groups or NH, NHCO. Why it is having? Because this NH group, it is having the this NH group having, it is increases, you can say the solubility, it is the basic groups, and it is having the lone pair of electrons, which is one of the hydrogen bonding domain, you can say. Similarly, the distal aryl ring, that is A with the R2 substitution, you can go for any. Here it is given chlorine, fluorine, you can go for another ones, but you have to put the different halogens. You try yourselves and go through the binding of different functional group to the aryl nucleus, which is attached to the quinoline. In all the cases, you just see that how the second position and the third position it is structurally modified to give the different derivatives of quinazole nucleus. But there is fourth position always there is presence of ketonic group. But I have substituted with the your ketonic group with the double bond S, thigh group also. By modifying, by having the phosphorus pentasulfide adding, we are getting the C double bond S. We want to see whether that particular group will increase the polarity or not, whether it is enhancing the stability of that particular quinazole nucleus, or whether it is having the biological activity, good or bad. So huge amount of you go for the structure and synthesis, then you will get the structural activity relationship. If you are getting the once you are getting the good SAR, then you continuously search it, so you will definitely get. And the another thing is that the pharmacokinetic profile. So many drugs are having the metabolic effect, and so many drugs are having the very less oral solubility, oral absorption. How do we can increase the oral availability of the drugs? By giving the bioavailability. Suppose you are going for the, you can say the anti-cancer drugs. So you will find there are so many bioavailability enhancer drugs are there. So you can add that bioavailability enhancer, which increases the bioavailability of that particular drug. So bioavailability enhancer also one of the recent topic in the research. So that part you can focus how you can increase the bioavailability with the marketed drug also. With the marketed drug, we can go for the bioavailability. For example, I am telling one drug is the allotinib or getinib. Their bioavailability, although they are clinically approved, but their bioavailability is less. That is why you can go for the some sort of naturals, or you can say the herbal bioavailability or bioenhancers you can add. So that is also one of the research. So one is the synthesis, one is the bioavailability enhancing, how you can increase the pharmacokinetic profile, that part you can include that. So you can go for the that one. So these are the various synthetic scheme targeted compound which we have done in our lab and which we have synthesized. So next is the drug designing studies that I have already told. So drug designing study in India, basically what we are doing. In the drug designing, we are doing the docking, we are doing the Schrodinger software using auto docking, XLRs, homology modeling, like that. There are so many softwares are there. We are going for the drug designing. But we are getting very, very less compounds, lead molecules. From that also, whenever you are coming to the wet lab, so this is the dry lab condition. You can say the wet lab, it is very difficult to synthesize. This is I'm telling the practicals. The practical thing in India, actually, it is difficult to synthesize the compound. Sometimes also what happens, whatever the drug, you will get the good result, the past prediction in the drug designing, but it is not coming to the practical results whenever you are analyzing or evaluating the biological activities. So that is also one of the huge problem with us in the laboratory. So whatever the designing drug is coming, it is not coming to the real biological activity. But what we have to do, we are need for the papers. That is why we are just simply manipulating. Okay, this is coming this kind of data. So this is the problem, the real scenario I am telling. So here the drug designing is the targeted compound. It is having the hydrophobic domain, hydrogen donor domain systems, distal aryl ring, hydrogen acceptor systems. So according to that, how the drug molecules bind to the particular receptors. So here they are binding to different receptor, and these are the various amino acid binding sites in the receptors. Remember one thing, whatever the process, you can go through that, but the basic process you should understand why it is happening. That is the most important part in case of the synthetic chemistry. So what we are doing, we are going for the, okay, the designing is there. The designing will do. We are getting the results. But the basic thing we should understand why it is happening and what is actually the structural activity relationship, what is the real mechanism behind it. That you have to understand, which is very much necessary in case of the research. If you do not understand of your particular research, then it is totally zero. So that you have to understand what I am going for the research. Suppose you are going for the designing, so you have to understand the basic things what I am going to perform. Because the basic process, all the process, it is already in the computers. That you put it and you will definitely get the results. Then why I will do, that is very much important. The rationality behind it, the justification, the objective, the aim should be perfect, so that you can get the result. It may take a lot of time, but you will definitely get a results. So that part you just go through analysis, my dear faculty. So you just segregate yourself into different parts. Designing one part, synthesis one part, characterization one part, and ecological part one part. Do not do not involve yourself in all the research. You segregate yourself or take the particular teachers, or you can say the research scholars, and do your part. So then only you can get the fruitful result. If you are doing the continuously going the research in the wet laboratory, so go continuously go through the schemes, various schemes you perform, then you will definitely get a result. And that particular result you can take and go for the characterization, and you can see the biological activity. So you have to go through that. So today the topic is about the quinazoline. Suppose you are taking, you can take the marketed available quinazoline also. In that marketed available quinazoline, you can directly add one prodrug also. So that is prodrug design also one of the recent concept product designs. For example, I am telling about the Swedish people. Suppose you are going through the solid lipid nanoparticle. So I have seen one of the drug is the nipacotic acid derivative. Nipacotic acid itself is one of the anti-epileptic agents. So if you go through the nipacotic acid, that is also one, you can go for the solid lipid nanoparticles. So you have to divide yourself. First, you go through the design, synthesis, activity, as well as the formulations. Then you go for the patent. It is very easy whenever you are segregating yourselves. If you compile it, no, I will do alone, or 2 to 3, it is very difficult to get the results. First, you segregate yourselves into different parts, then you go for the patent. Patent is nothing but you have to go for the segregation. The result you will get, continuously go through the result, definitely you will get the good, you can say the acknowledge in any fields, that ones. Next is the, recently I have just performed and some synthetic compounds in my lab. That is about the, these are the various quinazoline derivatives which are having the huge anti-epileptic activity. So these are the different compounds, 5 compounds. This I have already given. So these compounds for the applied, I have applied in the US patent. So just hope for the best. And it is not only for the anti-epileptic activity, I have also have that anti-hypertensive activity, because these are the compound which is having the just like the prazosin structure. So you don't go for the particular specific one. It can be serendipity. Suppose you are going for one particular activity, but it is showing the different. That is also very good. That you can see. So continuously research should be in a cyclic process, and it is continuous process. Divide yourself and do the work. Synthesis, one, characterization, and directly send to the lab. If you don't have the lab, you directly put anywhere else. These are the things, but it should be continuous process. Then only you will get the results. The problem is that suppose we are having the 5 compounds. In that 5 compound, we are going for characterization, but we are not getting. But characterization should be perfect. You should get the pure compound, and the activity may be 0. Suppose you are getting a good, not having the good activity, suppose in anti-cancer I am telling. But in anti-cancer you are getting the 0 result, but we are disappointed. Okay, 0 result is coming, no, we have to go for manipulation, we have to complete, we have to go for the paper. Don't be hopeless. If you are not having the good activity in that particular compound, send to others, go through the literature search for the SAR, then you can go for the other activities also, you can try yourselves. But don't be silly thought that if this activity is not coming, I will not go for another activity. So it should be a continuous process, all kinds of activity, whether it is anti-cancer. For example, I am telling about the quinazoline nucleus. So this quinazoline nucleus is having the anti-cancer activity, your anti-epileptic activity. You can correlate any one, or any 2, or any 3, it depends upon you. Go through the huge synthetic process, synthesize the compounds, characterize, and go for various pharmacological activity, whether it is in vitro model or in vivo model. But you have to see the structural activity, it is very important. And you can go through the QSR topic like Topless approach also you can see, and you can get the ideas from that. So divide yourself, you will get. So these are the various compounds that we are getting the some sort of antihypertensive activity, as well as these are having some sort of anti-epileptic and CNS depressant activities. So we are going through the continuous analysis, and these compounds we are getting the fruitful result. We are waiting for the results for that US patent. Let's see what happens. Synthetic scheme is there, and one thing I want to tell you, suppose these are the quinazoline derivatives. These are having the 3 steps or 4 step synthesis. But in that 3 step or 4 step synthesis, the reaction intermediate is the benzoxagenone derivative. So benzoxagenone derivative itself, they are some sort of anti-cancer activities. So you can put, my dear faculty, you can have the idea about the benzoxagenone and put that benzoxagenone in the mind and go through the various synthetic scheme for the benzoxagenone and go through the various activities. So that benzoxagenone, I have recently put one benzoxagenone with the pyrazoline nucleus for the anti-tubercular activity. So you can put that benzoxagenone with the pyrazoline for the anti-tubercular activity. You can go through the research that one. Similarly, my recent work is right now on the benzoxagenone, one part benzoxagenone with the quinazoline nucleus. I want to go for that. As I have getting good anti-cancer activity on the quinazoline, as we are having the research literature review that benzoxagenone is having the anti-cancer activity, so we are just compiling. We are just adding that two, and we are going for various synthetic scheme, various derivatives of quinazoline, benzoxagenone, and analyzing their antiviral activity as well as the anti-cancer activity. So that part you can see. So reaction intermediate is very important. For example, one of my research is on the pyrazoline nucleus. In masters I have completed. And in that pyrazoline nucleus, we are having your starting material is the chalcone. The intermediate, we are getting the chalcone derivatives. So that chalcone also having the MAO inhibitors. In masters, I have done the antidepressant activities, MAO inhibitors, and anti-epileptic activity on the pyrazolines. In that, I have get the chalcone derivatives. So that chalcone also, it is itself having the MAO inhibitor. That is non-selective, it is MAO. If you go through the research, you will get the chalcone is having the MAO inhibitors activity. So that also you can do it. It up to you, the reaction. Basically we are just omitting the intermediate part from our mind, that we don't go for the research any kind with the intermediates, but intermediate itself it's showing some sort of results. That one also you can see. And one of the important thing for a kind information, negative research, negative results also always you can say the appreciable, but we are not thinking, we are thinking negative result. No, we are going for the manipulation. We'll manipulate the data, we'll get the results. See, remember one thing, paper is the art actually. If you are writing the paper, it is always artistic. But the real field is the wet lab. So that you have to perform. Whenever you are going for the paper also, continuously you write the paper. Suppose you are completing the synthetic schemes, write down the synthetic part in the papers. But day-to-day activity, you should write in your one diary, maintain the diary, and day-to-day activity, whatever the results you are getting in the research every day, you have to particular day you write everything. Suppose you are having the TLC optimization, or you can say the solvent optimization, everything you put the data so that you can have the results. At last you can get the results. You will get one idea from that. So every day, whenever going for the research, you write everything in the diary. Suppose you are going for the synthesis, that part also you can see. Okay, today I am going through the this part, and every 1 hour or 2 hour I am going to analyze the RF value or TLC. So that you can also do you write in your diary and maintain that. At last you will get the result. So that is also one of the optimization, that is also one of the research. But we are thinking the research means we will get the particular lead compound. It is never. Research any kind, whether it is synthetic, whether it is optimization, whether it is designing, anything. You put yourselves continuously and do the research. Every day you can go. Suppose you are having the lab synthetic chemistry you are doing with the students. At the time also you can also put one reflux condensation synthesis continuously along with the students. You can go and you can just analyze, discuss that particular thing, whatever you are going the research, that part you can discuss with the students also. So teaching a research are always correlated, but we are thinking teaching a research completely different. No. It is always there. Research lab is something different, but in the research lab, in the academic institution, it is something different. Whatever you are doing, it should be always practical, whatever you are doing, and you can show this to the students so that you can have the knowledge, they can have the knowledge. For example, I am telling the quinazoline nucleus. You are going through the tremendous literature reviews. If you are going through the tremendous literature review, obviously you will have the idea that what are the different, what kind of activity, what is the synthetic scheme, what is the heteroatom pairing. Basically, you can analyze, you can establish yourself as a teacher to the students by rearing the knowledge, these kinds. So these you can do in the academic institution. Faculty development program, it should be practical. But whatever we are doing, we are just going for we are showing our works. So faculty development, it should be very much qualitative, my dear faculties. Whatever you are doing your lab, it should be perfect. Go through the practical analysis in your lab whenever you are going from to the labs. along with the students. Simultaneously you work. Then only the student will believe you, okay, sir is doing the research, we will also do. They will be inspired through you. So that part you can do it. Each and every day you go for the research, whether it is half an hour or 1 hour or 2 hours or 3 hours. So I will directly request to our principal, usually the Dean of Mpa Pharmaceutical Science College and Research Center, sir, please provide the time to the teachers, extra time they can go for the research. And one of the thing is that we are having the duty, suppose 9 to 5 works. But do not hesitate to go for 1 hour, 2 hour research every day. You should be present 1 hour, 2 hour extra for your practical work every day. And whatever the practical work you are doing, you put in the diary and maintain this continuously, then you will inspire yourselves. Okay, I am doing continuously and go through the research. Every day you just do it, then you will definitely get a good results. And your paper and your articles, so whatever the research, you will definitely get a patent. So our main target is what? Good papers, good patent, or good book, whatever. But you should be targeted that whatever I am doing, I should be thorough about that. That I am telling. So next with this, I have told the new areas of research. So new areas of research, it is about the quinazolid nucleus and the molecular hybridization with the chalcones derivatives. That is one of the that I am doing. So how the chalcones, actually the quinazolid nucleus, they are having the anti-cancer activity and the antiviral activity. So that we are also performing, but we are not, till now we are under the processing stage. We are optimizing various derivatives of that chalcones as well as the your quinazolid nucleus. So optimization is very important. In my research, in my master's and PhD, I have given 5 to 6 months for the synthesis, 5 to 6 months only for the optimization, not for the continuous bulk amount of drug synthesis, for the optimization. Whether it is solvent optimization, whether it is temperature optimization, whether it is the amount, whether it is the TLC, you can say analysis, and whether it is getting the pure compound through the flash chromatography techniques. So these are the things we are doing. So it is very much important. Do not compare yourselves. The academic institution should not be compared with the various research sections. For example, if you compare with the various CSR lab, it is the hell and hand difference. But whatever the research we are doing, you can start from a very small thing. You can go through the, you can say the metal complexes, and suppose you are taking the copper. So how you can go for the copper coordinating compound? That part also you can do it, but small synthesis you do it. Definitely you would get the results, and through that you can have the good projects. So whatever you are getting the result, through that you can have the good project in various project. Those who are the different government and private government agencies, private agency who are giving the distribute the project, that you can also apply. So that you can do it. Collaborative research is very important for all of you, because in the academic institutions, very less instruments are there for the analysis. So that if you go for the collaborative research with the various academic institutions as well as through the various research lab, or you can go through the you can say the various, suppose twinning project I am telling about, the DBT or DST. We have already applied twinning project. I have already applied 3 projects, DBT and DST project, twinning projects. So that should be intercontinental basis, you can say, suppose in India or you can say the Sri Lanka you are applying. So that part you can do it. So twinning project or the collaborative research is very important. Until unless it is means you are not collaborating, you will not get the result. For example, your synthetic lab is good, but the activity lab is not there. But the characterization we have to send other thing. If you collaborate yourself, then only you will get the definitely getting the results. So you should go for the collaborative research, my dear faculties. In yourself you can do it. You just go through the, suppose you are going for the publishing one papers. At the time also you go for the divide yourself, that as I have told that you have to go for the synthetic one part, activities one part, that part you can do it. So what, suppose for example here I am telling about the my quinazolid, whatever I am the anti-cancer lab, I am just having that I am doing the anti-cancer activities, but it is very difficult to maintain the cell lines. So what we are doing, we are MOU tie up with the Institute of Science Technology in Guwahati, ISST. So through that only our faculties and me, we are also going to that maintaining our cell lines. Suppose I'm telling about the HeLa cells or Dalton cell lines. So through that we are going through the research. We are synthesizing this one in our lab, and we are directly sending this characterization part to others who are having the good facilities and the activities one part. So then only you will get the result. We are also getting the result from my side. This is practical I'm telling. So you should go through that once. So these are all about this. So from that my 12-year experience, I have already researched and reviewed 30 and 5 papers through my various ex-students. We are going for the already applied to EJMC and as well as the chemical paper springers. Two of my papers, we have got the top hottest articles, top 7 positions we have got already in the year 2010. And I have book published, it is 5. One is in the processing conditions, and book I have published 4 about the C, in which here I want to tell you one thing. Suppose in the research, you are having the huge amount of literature review of that particular quinazolone. So one book I have published on the quinazolone nucleus, one book directly, because more than 100 or 200 pages, more than 100 pages I have got the 100 articles I have got the literature review. So I thought that if I'll put as a book, it will be obviously the research student will get the about the literature review. So that's the thing. So if you are having the good literature review, you can publish as a book like that. That you can do it. And patenting, I have already 4 patents, so 3 is already granted and US patent is applied. So that US patent I have in the synthetic chemistry. That Australian patent is along with my all BHU batchmates. So that is about the some herbal formulations. South African patent, it is also about that. It is already almost done. So it is about the one of the vaccinations actually recently coronavirus. So that part we are doing. And German patent also it is on the synthetic chemistry. So that part I am doing continuously. You have to engage yourself research, then definitely you will get one patent. And awards and recognition like that, as already Nismet has told about me. So I have got qualified the GATE, GPAT and NET also in life sciences. And I have also best teacher award excellency towards teaching contribution in Girijananda Chaudhary. Then I got one outstanding scientist awards in the northeastern regions. Got one RPS project 24 lakhs. See, in the project in my starting of my career, so what happens, I have already applied 3 to 4, like CSIR I have applied, DST I have applied, but it is cancelled. Only one, this RPS project, AICD 24 lakhs I have got. And one departmental project were also our college Sriman Sankar Academy Society, they have given 1 lakhs for the research in antiviral drugs. And already I have applied 3 projects, applied 5 crores under various formulations, as well as from the chemistry also DBT and DST research by twinning, by the collaborative ones. Then some workshop I have attended like 30-35 guest lectures given, FDP speakers I have already given 10, and the GPAT seminar. Actually I am a GPAT coach also all over India. I am giving the coaching to GPAT, 25 I have attended. And throughout my career, so I have given the more than 1,000 students all over India, they are getting the GATE and GPAT. So if any student require any kind of things from me, so that part also I will give from my sides. If anyone want to, if your college, if the students are interested through that you can also have the seminars for that. So this is my contact numbers, and you can have the contact if you want to genuinely interested for any kind of research or any kind of review articles, research article writing, project writing, how you are going for the project writing and patented also. So that part you can, it is you can send anything to my email or you can contact me through the WhatsApp also. So we can go for that. Our sites, we are interested from the NEPAD group of institute for the collaborative research. If you are interested, so that part we will do it. Next is the acknowledgement. I express my heartily thanks to the chairperson, Dr. Muhammad Guljar Ahmad sir, principal and dean, Yanapia Pharmacy College and Research Center, Yanapia deemed to be University, Mangalore. Then I owe special thanks to Rokia Sultana madam under the professor and head department of pharmacognosy. Special thanks to Mr. Saurabh Kumar Mahata, assistant professor, department of pharmaceutics. And at last, I express my sincere thanks to the team of the organizers for organizing this very beautiful, you can say the FDP programs. So thank you so much. So thank you. These are the two questions. These are the two questions.
Sir?
Hello.
Yes, sir. You can turn off your slides.
Okay,
I'll
so
connect it
actually some
later.
sound is coming. Okay, so these are the two questions that I have given. So telempanel is dash derivatives like your quinazoline, benzodiazepine, thiodiazole, oxadiazoles, and MPA. I have given this neurotransmitter whether it is inhibitory or excitatory or it is both or none. So these are the two questions. So thank you so much.
Yes, sir.
So if anybody is any question, you can ask me. So it is
Yes,
right
sir.
now the questionnaires timing.
Yeah. Thank you, sir, for your sharing your knowledge. It will be a great pleasure for us to listen to you again in our future. I hope that each of the attendees now have a deeper understanding of heterocycles and neuropharmacology. The session is now open for question and answers. First, I'll be addressing the speaker with the queries posted in the comment section. Further, if anyone wishes to directly interact with the speaker, are requested to raise their hands by clicking the icon seen on the Meet screen. We would call up your names and you can unmute yourself and clarify your doubts. Once your queries are addressed, you are kindly asked to raise down the hand by re-clicking the icon. So here we have the first question from Zial Haq. Which methods or software are used for the study of receptor antagonism?
Yes, actually, I am taking the Mpa receptors. So I am Mpa receptors. I am taking which the drug will bind to the non-competitive sites, and I am taking the branded drugs like telempanel, perampanel, and the quinazoline derivatives. So that Mpa receptors we are taking, which is non-competitively binds. So Mpa induced seizures we are taking.
Hope Zial Haq, you have got your answer. We have the next delegate, that is Gunji Venkateshwaralu, who have asked, if you want to publish book from review articles, we require permission from them. How is it?
Definitely you have to write if you want to publish one paper, so publish one book articles, you have to cite them. The citation is very important. Before that you have to acknowledge them, and one of the thing is that you have to mail corresponding mail you have to send them, whether you are they will approve or not. It is very important because the copyright is along with them na, because the citation they need actually, so you have to go for the citation whenever you are publishing that. That's very important. So you have to go through that one. So whenever you're publishing the review papers as a book, so by taking their permission only it is possible.
I hope, Gunji sir, you have got your answer. We have the next question from Dr. Avneet Kaur. Can thesis be printed in the form of book?
Yes, definitely. Your thesis, it is, you can print in the form of book. It is up to, because it is the copyright by you only, and it is by yourself. So you can go through the book publications you're printing, because it is the one of the research. Na? Research, it can be printed as book. Definitely you can do it. It's good actually.
So we have a connecting question from Ziol Haq. What about software? Which one we can use for VLife, Autodoc, or anything else?
Yes, actually, for the receptor, actually, so first here I am doing the Autodoc actually. So in the quinazolines, we have taken the Autodoc directly. So the Autodoc, you can do it. Through the Autodoc process, we are having the different receptors you can get. So that receptor, actually, I have forgot the name. So that receptor, we have downloaded from the PDB databanks. Through that, we are binding the ligand drug binding interaction we have studied. Autodoc, you can do it, no problem.
Hope Ziol sir, you have got your answer. So these were the questions that were been posted in the chat box. If anyone else would like to ask any questions directly to the speaker, you're free to raise your hand. We have Bargi Jain who have raised her hand. Bargi Jain, you can unmute yourself and ask the question directly to the speaker. Bargi Jain. Yes, I hope it is an accidental click on the hand raised.
It's
So
okay.
we'll
It's okay.
continue
Okay.
or conclude this session. Thank you,
Okay,
sir,
so
for
let me
sharing
let
your...
me say, Nismita, one thing I want to see, so many delegates they have just given acknowledgement. Thank you, sir. So thank you so much, everyone, for listening to me. You have bothered me a lot of 1 hours. So it is, I think, hope you can understand the practical things. It is very important. You should go through the practicals because in India the basic problem is the research. We are thinking a lot, but execution is very less. The problem is that the laboratory facilities we don't have the practical things. We are thinking a lot, but the execution is very less. So you have to plan accordingly. Whatever the instruments, anything the facility is there, whether it is dry lab or wet lab, according to that, perform yourselves and go accordingly. Thank you so much.
Thank you, sir, for sharing your vast expertise with us. I deem it a great honor and privilege to propose the vote of thanks on behalf of Staff Welfare Committee, Mpa Pharmacy College and Research Center. On behalf of YPCRC, my sincere gratitude goes to today's speaker, Dr. Bishwajit Dash, Professor and Principal of NEPDS College of Pharmaceutical Sciences, Assam, India, for enlightening us on heterocycles and neuropharmacology and how collaborative research is important in getting a good result and research. My special thanks to Dr. Mohammad Gulzar Ahmed, Dean and Principal of Anapoya Pharmacy College and Research Center, for providing us immense support. This event would not have been possible without the efforts of the organizing team, the Staff Welfare Committee, YPCRC. I would like to thank Dr. Rokeya Sultana, Professor and Head, Department of Pharmacognosy, for her foresight in organizing this FDP as well as the members of the Staff Welfare Committee. I would like to express my gratitude to the Anapoya deemed to be university management as well as all of the teaching and the non-teaching staff of YPCRC for their guidance and assistance. Last but not the least, I would want to express my gratitude to all the participants from across the world for attending this afternoon session. Dear delegates, please fill out the feedback form for today's session, which is already been pasted in the chat box and kindly submitted. Thank you one and all. Have a nice day.
So Nismita, so Surai left right now. Can I?
Yes,
So you
sir.
can
Yeah,
you
you
leave
can
them.
leave, sir.
Okay, okay, okay.
Any further communication will communicate to you directly, sir.
So dear participant, you can leave the meet.